In this lecture, we'll review the fundamental features of molecular biology. The goal is to understand the overall framework of information storage and transfer in living cells. This overview should give us a better understanding when we go back and study each stage of genetic information transfer in greater detail. We review this slide to remind us that there are some problems to be overcome in the process of DNA replication. We've seen that DNA replication is semi-conservative. The semi-conservative model requires that the two parental strands of DNA must be completely separated in the process of replication. Those antiparallel strands are joined by the hydrogen bonding of the base pairs. All of those hydrogen bonds must be broken before replication can occur. In addition, the two parental strands are physically wound around each other in a helical structure. In order for replication to proceed, the two strands must be completely unwound and separated. In this slide, we review the basic structure of DNA. This tetranucleotide DNA sequence contains the four constituent bases of DNA: adenine, cytosine, guanine, and thymine. The nucleotide units all contain deoxyribose, and they're all linked by 3', 5' phosphodiester bonds. The way this structure is written implies that it's a segment of DNA that continues on in each direction. The phosphate residue at the upper left would lead on toward the 5' end of the molecule, whereas the oxygen atom at the lower right would lead on to the 3' end. Base pairing refers to two nucleotides on opposite complementary DNA or RNA strands that are connected by hydrogen bonds. Those are called a base pair, often abbreviated BP. In the Watson-Crick model for DNA base pairing, adenine A forms a base pair with thymine T, and guanine G forms a base pair with cytosine C. In RNA base pairing, thymine is replaced by uracil. Adenine forms a base pair with uracil, and guanine forms a base pair with cytosine. Hydrogen bonding underlies base pairing. Appropriate geometrical correspondence of hydrogen bond donors and acceptors allows only the right pairs to form stable interactions. Base pairing occurs between complementary bases in DNA and does not occur between non-complementary bases. The complementation between those base pairs represents the most important example of hydrogen bonding in living systems. Base pairing is at the heart of DNA replication, of transcription of DNA to RNA, and of translation of RNA into protein. The diagram of an AT base pair shows that the complementation between these two bases involves the formation of two hydrogen bonds. One hydrogen bond occurs between the hydrogen atom of the 6-amino group of adenine and a carbonyl oxygen atom of thymine. A second hydrogen bond occurs between one of the ring nitrogen atoms of adenine and a hydrogen atom attached to a ring nitrogen atom of thymine. For the AT base pair, the distance between the 1' carbon of the deoxyribose of one chain to the 1' carbon of the deoxyribose of the other chain is about 1.11 nanometers. We've just seen that the pairing between A and T occurs at a distance of 1.11 nanometers. The distance for the GC base pair is essentially the same, 1.08 nanometers. Both the AT and GC interactions involve a purine interacting with a pyrimidine. If a purine bonded to a purine, the bond distance would be greater because purines are larger than pyrimidines. If two pyrimidines were bonded, the distance would be correspondingly smaller. Because base pairing always involves a purine bonding to a pyrimidine, the strands of the DNA double helix remain at a constant distance from one another. There are 3 hydrogen bonds in the GC pair. A carbonyl oxygen of guanine bonds to a cytosine amino group. A hydrogen attached to a nitrogen atom on the guanine ring bonds to a nitrogen atom on the cytosine ring. An amino group of guanine bonds to a carbonyl oxygen of cytosine. Collectively, the energy of these 3 GC bonds is greater than the total energy of the 2 AT bonds. Traditionally, this has been taken to reflect the greater number of bonds in the GC pair. However, recent data suggests that other factors may play a role in the collective affinity of these base pairs. In any case, it would take more energy to separate two strands of DNA from a polymer with GC bonds than it would to separate strands from a similar AT polymer. The ball and stick model shows three base pairs of DNA. You can see in this diagram that the individual bases are planar, but the base pairs are slightly tilted with respect to each other. In addition to the energy of hydrogen bonding, the DNA helix is stabilized by the energy of aromatic stacking between successive bases in the helix. Now we come to the infamous central dogma of molecular biology. It's important to remember that the central dogma of molecular biology concerns the flow of information from DNA, the hereditary storage of genetic information, to RNA, and then from RNA to protein. This means that the arrows in this scheme do not signify the chemical conversion of one compound to another. Rather, DNA serves as a template for the synthesis of new DNA and also of RNA, and RNA provides a template for protein synthesis. The semi-conservative model of replication tells us that in the first generation of DNA synthesis, each daughter molecule of DNA contains one parental strand and one newly synthesized strand of DNA. Initially, there's one DNA molecule containing two parental strands. After one generation, there are two progeny DNA molecules. Both of these molecules have one of the original parental DNA strands and one newly synthesized strand. After two generations, there are four DNA molecules. Two of the DNA molecules have one of the original parental strands and one newly synthesized strand. The other two DNA molecules have two newly synthesized strands. If further generations were generated, there would always be two DNA molecules with one of the original parental strands and one newly synthesized strand. All the rest of the DNA molecules would contain only the newly synthesized strands. The figure on the left shows that the absorbance of light by DNA is less for double-stranded DNA than it is for single-stranded DNA. This change in absorbance can be used to monitor the melting of double-stranded DNA. The figure on the right shows a melting curve for DNA. As the temperature is raised, the hybridized strands begin to separate. The midpoint of the curve where the DNA strands are 50% separated is defined as the melting temperature, Tm. At an elevated temperature, the strands will be completely separate. Upon cooling, the strands will rehybridize to form a double-stranded product. It turns out that single-stranded DNA can be hybridized to complementary DNA or complementary RNA. The DNA hybridization technique can be used experimentally with any set of nucleotide segments that have significant amounts of complementary base pair structure. The base pairing does not have to be perfect for the hybridization to occur, but there must be a significantly high percentage of matching base pairs. Base pairing can occur between two distinct DNA strands, or between a DNA strand and a complementary RNA strand. This slide shows that single-stranded DNA or RNA can also exhibit base pairing. The figure on the left shows a stem loop structure for single-stranded DNA. The bases in the stem are complementary and hybridized to form base pairs. The bases in the loop are not complementary and do not hybridize to each other. The figure on the right shows that similar stem loop structures can be formed within single-stranded RNA. Here's an example of base pairing within an extensive region of RNA. There are long complementary regions that contain occasional mismatches and bubbles. There are also a variety of stem loop structures which branch off from the main chain. DNA replication is the process by which parental DNA is copied. The general pattern of DNA replication involves the formation of two copies of progeny DNA from one copy of parental DNA. The process of replication is semi-conservative. DNA polymerase is an enzymatic complex that is responsible for DNA replication. DNA polymerase catalyzes phosphodiester bond formation. The DNA polymerase complex catalyzes the step-by-step addition of deoxyribonucleotide units to a DNA chain. The new DNA chain is assembled directly on a pre-existing DNA template. The reaction catalyzed is DNA with N number of nucleotides plus dNTP yields DNA with N plus 1 number of nucleotides plus inorganic pyrophosphate. The template for replication can be single-stranded DNA or double-stranded DNA. If the parental DNA is double-stranded, one of the chains must be broken at one or more sites. If the parental DNA is single-stranded, the template DNA must be bound to a primer strand having a free 3' hydroxyl group. The DNA polymerase reaction also requires all 4 activated precursors, DATP, DGTP, DTTP, and DCTP, as well as magnesium. The polymerase reaction adds nucleotide units to the 5' end of the DNA primer strand. In the process, the deoxynucleotide triphosphate substrates are cleaved to release pyrophosphate and to add a deoxynucleotide monophosphate unit to the growing strand of DNA. The added nucleotide is complementary to the template DNA nucleotide. The synthesis of DNA proceeds from the 5' end toward the 3' end. The reaction mechanism involves the terminal deoxy residue on the primer. The 3' hydroxyl group of the terminal deoxyribose attacks the inmost alpha phosphate residue of the incoming deoxynucleotide triphosphate. The phosphodiester bridge is formed linking the primer to the new nucleotide unit. The 3' hydroxyl group of the new nucleotide unit can then attack the next incoming deoxynucleotide triphosphate. 3 types of RNA are involved in gene expression. Messenger RNA is the template for protein synthesis or translation. A messenger RNA molecule may be produced for each gene or group of genes that's present in E. coli. A distinct messenger RNA is produced for each gene in eukaryotes. The messenger RNAs comprise a heterogeneous class of molecules. In E. coli, the average length of a messenger RNA molecule is about 1.2 kilobases. Transfer RNA carries amino acids in an activated form to the ribosome for peptide bond formation. The sequence in which activated amino acids are polymerized from transfer RNA is dictated by the messenger RNA template. There's at least one kind of tRNA for each of the 20 amino acids. The tRNAs are relatively small, consisting of about 75 nucleotide units. Ribosomal RNA constitutes the major component of ribosomes. The ribosomal RNAs are relatively large molecules that play both a catalytic and structural role in protein synthesis. In E. coli, there are 3 kinds of ribosomal RNA called 23S, 16S, and 5S RNA. One molecule of each of these species of ribosomal RNA is present in each ribosome. The table shows that the 3 ribosomal RNA molecules constitute the largest fraction of cellular RNA, about 80%, and contain about 3,700 bases for the 23S, 1,700 bases for the 16S, and 120 bases for the 5S component. The transfer RNAs constitute about 15% of the total RNA and average about 75 bases, making them the smallest RNA molecules. The messenger RNAs represent about 5% of the total and have an average size of about 1,200 bases. The term kilobase is a unit of length equal to 1,000 base pairs for double-stranded DNA or 1,000 bases for single-stranded nucleotides. The RNA polymerase reaction requires the following components. A template is required. The preferred template is double-stranded DNA. Single-stranded DNA can also serve as a template. RNA, whether single or double-stranded, is not an effective template, nor are RNA-DNA hybrids. Activated precursors are also required. All 4 ribonucleoside triphosphates, ATP, GTP, UTP, and CTP, are necessary. A divalent metal ion to interact with the nucleotides is also required. Either magnesium or manganese is effective. The synthesis of RNA is like that of DNA in several respects. First, the direction of synthesis is 5' to 3'. Second, the mechanism of elongation is similar. The 3' hydroxyl group at the terminus of the growing chain makes a nucleophilic attack on the innermost phosphate of the incoming nucleoside triphosphate. Third, the synthesis is driven forward by the hydrolysis of pyrophosphate. The synthesis of RNA differs from that of DNA in that RNA polymerase does not require a primer. RNA polymerase lacks the nuclease capability used by DNA polymerase to excise mismatched nucleotides. The mechanism of RNA synthesis is virtually identical to that of DNA replication. The RNA synthesis proceeds from the 5' end toward the 3' end. The reaction mechanism involves the terminal ribose residue of the primer. The 3' hydroxyl group of the terminal ribose attacks the endmost alpha phosphate residue of the incoming nucleotide triphosphate. The phosphodiester bridge is formed, linking the primer to the new nucleotide unit. The 3' hydroxyl group of the new nucleotide unit can then attack the next incoming deoxynucleotide triphosphate. The terminology of DNA with respect to transcription is: the coding strand of DNA is not used for transcription, has the same sequence, except DNA has T instead of U, as the RNA transcript. The coding strand runs in a parallel direction to the RNA. The template strand is used for transcription, has the complementary sequence to the RNA transcript, and runs in an anti-parallel direction to the RNA. Transcription is initiated on the DNA template starting downstream from promoter sites containing consensus sequences. Consensus sequences are similar to each other in base composition and sequence, but an individual promoter sequence may vary from the consensus sequence by 1 or 2 bases. Prokaryotes such as E. coli have a TTGACA sequence minus 35 bases upstream and a Pribnow box minus 10 bases upstream from the transcription start site. Eukaryotes have a CAAT box minus 75 bases upstream and a TATA box minus 25 bases upstream from the transcription start site. Transcription is terminated by various signals. In E. coli, a stable hairpin followed by a sequence of U's leads to dissociation of the RNA from the template strand. In eukaryotes, messenger RNAs are synthesized in the nucleus and then transported out into the cytosol where protein synthesis takes place. The messenger RNAs are modified in the nucleus before being exported. A nucleotide cap is added to the 5' end and a poly A tail is added to the 3' end. Amino acid-specific tRNAs carry and donate activated amino acids during protein synthesis. In the example shown, the alpha-carboxyl group of the amino acid is attached to the 3' hydroxyl terminal group of the transfer RNA by an ester linkage. The amino acid is esterified to the 3' hydroxyl end of the transfer RNA. A triplet anticodon on the transfer RNA hybridizes to the complementary triplet codon of the messenger RNA. Amino acids are encoded by groups of 3 bases, triplets, in messenger RNA, starting from a fixed point. 3 nucleotides encode each amino acid. The code is non-overlapping and has no punctuation. The code is degenerate for some amino acids. That means that more than one triplet codes for certain individual amino acids. The codes UAA, UAG, and UGA are termination signals that stop protein synthesis. The triplet AUG is a start signal as well as a code for internal methionine residues. There are 64 possible codons that can be generated with 4 different bases, combining in any order to form triplets. It turns out that 3 of these are nonsense or stop codons, which code for the termination of translation. That is, they don't code for any amino acid, and when the translation machinery reaches one of these three codons, translation ceases. You should know these stop codons, which are UAA, UGA, and UAG. You should also know the codon for methionine, which is AUG. That methionine is generally the first amino acid to be incorporated into a protein. Mercifully, we'll spare you learning the rest of the code, unless you really want to. The way this matrix works is that the first letter, 5' position, of the triplet codon is given on the left side of the box. The second letter of the codon is on top of the box, and the third letter, the 3' position, is on the right side. For all three letters of the triplet, the bases are arranged in the order UCAG. In prokaryotes, the initiation of protein synthesis occurs downstream from a purine-rich Shine-Dalgarno sequence on the messenger RNA. This sequence binds to a complementary region on the 16S component of the 30S ribosome. The first AUG codon downstream from the Shine-Dalgarno sequence is recognized by a transfer RNA carrying informal methionine. In eukaryotes, the initiation of protein synthesis occurs at the first AUG sequence that occurs downstream from the 5' cap. That first AUG codon is recognized by a transfer RNA carrying methionine. Many eukaryotic genes have introns, intervening sequences of DNA within the gene that don't code for amino acids in the final protein product. In genes that contain introns, the coding regions are referred to as exons, express sequences. The beta-globin gene shown here has 2 introns. During transcription of eukaryotic genes that contain introns, the entire gene is transcribed into messenger RNA in the nucleus. The primary transcript is then modified with a cap and a poly-A tail, and then the introns are removed. This process is called splicing. When those operations are completed, the mature messenger RNA is ready for export to the cytosol. Consensus sequences of GU at the 5' end of the intron and AG at the 3' end delineate the points at which splicing occurs. At these sites, the intron is cut out and the ends of the exons are spliced together. The exons in split genes often code for distinct domains within proteins. The occurrence of introns within eukaryotic genes may facilitate gene shuffling by allowing various domains to be recombined. This type of gene shuffling is of evolutionary advantage because by gluing different functional regions together, it can create new proteins. What is a genome? A genome is the complete sequence of an organism's genetic information. The human genome is the complete sequence of all 23 human chromosomes. The human genome contains about 3 × 10⁹ base pairs. Information about the complete structure of a genome can provide a potential source of knowledge about the proteins that an organism can synthesize. However, the information about genomes is not easy to interpret, and it works the best when the genome structure of one organism is compared to the genomes of other organisms. At this time, there are many complete genome sequences available, along with sophisticated computational techniques for assessing the functions of individual genes. There's a dramatic difference between prokaryotic and eukaryotic gene structure. Prokaryotic and viral genomes are very efficiently organized. They contain wall-to-wall genes with very little wasted space. In contrast, eukaryotic genomes have a relatively small fraction of the DNA devoted to genes. Most of the genome in higher eukaryotes consists of repeated DNA of various classes. There are simple sequences of 10 to 12 base pairs that are repeated millions of times. Short interspersed nuclear elements, SINES, S-I-N-E-S, are short DNA sequences, less than 500 bases, that represent reverse transcribed RNA molecules originally transcribed by RNA polymerase III into tRNA, rRNA, and other small nuclear RNAs. SINES do not encode a functional reverse transcriptase protein and rely on other mobile elements for transposition. The most common signs in primates are called ALU sequences. ALU elements are 280 base pairs long, do not contain any coding sequences, and can be recognized by the restriction enzyme ALU1, hence the name. With about 1,500,000 copies, signs make up about 11% of the human genome. The distribution of these elements has been implicated in some genetic diseases and cancers. Long interspersed nuclear elements, or lines, are a group of genetic elements that are found in large numbers in eukaryotic genomes. They are transcribed, or are the evolutionary remains of what was once transcribed, to an RNA using an RNA polymerase II promoter that resides inside the line. Lines code for the enzyme reverse transcriptase, and many lines also code for an endonuclease. The reverse transcriptase has a higher specificity for the line RNA than other RNAs and makes a DNA copy of the RNA that can be integrated into the genome at a new site. Because lines move by copying themselves, they enlarge the genome. The human genome, for example, contains about 500,000 lines, which is roughly 17% of the genome. There are also highly expressed genes present in several hundred copies. For example, the cell needs a lot of ribosomes, so it has 200 copies of ribosomal RNA genes. The human genome has a very high percentage of DNA that doesn't encode any genes. What's the purpose of that non-functional DNA? Some of it represents remnants of ancient gene duplications. Some of it is so-called selfish DNA. These regions may represent raw material for the evolution of new genes. One final series of Port Angeles pictures. The sailing boat is anchored in the harbor. Looking at this scene makes me want to sail off into the sunset. These are two of the tall sailing ships that visit Port Angeles frequently. Here they're out on a little harbor tour. Here are some of the creosote-treated pilings that support the local pier. The reflections in the water form rhythmic patterns. This raccoon is looking for his lunch under the pier. There's an abundance of shellfish for his dining pleasure. This is the Coho ferry that shuttles passengers and cars between Port Angeles and Victoria. Every spring there's a kayak symposium in the Port Angeles harbor. Kayaks are on display and one can sign up for kayak lessons or tours. My friend Bob sells produce at local markets or wherever people congregate. He has this great border collie that always accompanies him. A local artist forged this pair of cormorants. They found a permanent home near the city pier. We spend a lot of time near the local airport because my grandson plays baseball in a nearby field. The interplay of the hangars and the mountains yields a restful scene at twilight. This building was constructed in 1921 as a Masonic temple. It currently serves as a Masonic lodge and also as a location for all sorts of civic events such as dances and bingo games. This berry-laden vine is growing up the side of the First United Methodist and Congregational Church. These red flowers are part of a display on the waterfront. This photo was taken on First Street